most of the ocean is dark. Below about a kilometer, sunlight fails entirely. About 3/4 of the animals that live down there make their own light, through a single family of chemical reactions that has evolved, independently, several dozen times. The process is called bioluminescence. The light is produced when a substrate, generically named luciferin, is oxidized by an enzyme, generically named luciferase. The reaction releases almost all of its energy as visible photons and almost none as heat, which is why firefly light is sometimes described as cold light. In the sea, the emission is overwhelmingly blue-green, in the 440 to 480 nanometer range. Blue-green travels further through sea water than any other visible wavelength, and the animals that evolved to use it are the ones that were seen by others or could see themselves. The reaction has been invented, in the course of evolution, at least 40 separate times in animals, and by a recent estimate as many as 94 times across all life. Bacteria, dinoflagellates, cnidarians, crustaceans, mollusks, fish, and even one known shark family, the dogfishes, all produce light using their own chemistries. The chemistry is simple to state and intricate to perform. The luciferin molecule, the light-producing substrate, comes in several different forms: firefly luciferin, coelenterasine in many marine invertebrates, bacterial luciferin in luminous bacteria, and others. A matching luciferase enzyme catalyzes the oxidation. Energy is released in the form of a single visible photon per reaction cycle. The color of the light, green in fireflies, blue-green in jellyfish, yellow-green in bacterial colonies, is tuned by the specific enzyme and the substrate. In some animals, the light is produced by the animal itself. In others, like the Hawaiian bobtail squid, a specialized organ houses a culture of luminous bacteria that live and reproduce inside the host. A great deal of what we know about where bioluminescence occurs in the sea came from the work of the American biologist Edith Widder. From 1989 to 2005, she directed the Bioluminescence Department at the Harbor Branch Oceanographic Institution in Florida. Her 2010 review paper in Science remains the standard reference for the question of how widespread the phenomenon actually is. That 76% figure, the proportion of main taxa in the deep pelagic eastern Pacific that produce light, is from her work. In 2005, she founded the Ocean Research and Conservation Association and designed an observing platform called the Eye in the Sea, a deep-sea camera that watches passively in dim red light, emitting no white light of its own, so the animals behave naturally in front of it. In 2012, working with Tsunami Kubodra of the National Museum of Nature and Science in Tokyo, she captured the first-ever footage of a live giant squid, Architeuthis dux, in its natural deepwater habitat. A second scientific anchor came from much further back. In 1962, the Japanese chemist Osamu Shimomura, working at Princeton University with summer expeditions to the Friday Harbor Laboratories in Washington state, isolated two proteins from the crystal jellyfish, Aequorea victoria. One, which he called equorin, produces light in the presence of calcium. The other, which glowed green under ultraviolet illumination, he named green fluorescent protein. Decades later, the American biologists Martin Chaffee and Roger Tsien showed that green fluorescent protein, inserted into the genome of any organism, would cause that organism to glow visibly wherever the inserted gene was expressed. In 2008, Shimomura, Chaffee, and Tsien were awarded the Nobel Prize in Chemistry for the discovery and development of green fluorescent protein. A protein in a jellyfish had become the universal tracer of modern cell biology. The uses of the light in nature are varied. Dinoflagellates flash when disturbed. The bioluminescent bays of Puerto Rico, Jamaica, and Australia are produced by a single species, Pyrodinium bahamense. Deep-sea fish hang a bacterial lantern above their heads or in their bellies to attract prey. Ostracods and some squid release a cloud of light to confuse a predator, the marine equivalent of smoke. The counter-illumination strategy, producing light on one's underside that matches the faint downwelling daylight, erases the silhouette of an animal seen from below. Anglerfish, firefly, squid, krill, and many midwater shrimp use this form of active camouflage. The light is, in each case, an information channel, a signal, a trap, a disguise, or a warning. The field-defining review paper, by Stephen Haddock of the Monterey Bay Aquarium Research Institute, Mark Moline, and James Case, appeared in the 2010 Annual Review of Marine Science. Haddock and colleagues summarized, across 43 pages, the ecological, chemical, and evolutionary dimensions of the phenomenon, and emphasized how much remains unknown. For much of Earth's ocean, below about 1,000 meters, bioluminescence is not a curiosity, but the default medium of visual communication. Sunlight stops, organisms keep signaling. They do it with chemistry that emerged, by best current estimate, more than 500 million years ago, predating the Cambrian explosion, predating every vertebrate, predating every land plant. The earliest evidence of the trait appears in octocorals, a group of soft corals whose lineage is older than most of the phyla that followed. The reaction that lights a modern firefly's abdomen and the reaction that lights a dinoflagellate's cell wall are not the same reaction. They are convergent solutions to the same problem. Light carries information in water, and evolution has answered the problem independently many times. Most of the light on Earth is not sunlight and not firelight. It is the output, trillions of events per second, of a simple chemical reaction running in the cells of small marine animals in the deep ocean, where no human eye is watching.